everyone. My name is Nick Pena. I work in Dr. Daniela Zarnescu's laboratory in the Department of Molecular and Cellular Biology, and the title of this presentation is Uncovering the Synaptic Proteome with Fruit Flies. So what we normally kind of think of or are taught what a cell looks like is this basic looking spherical kind of blob where all these organelles are nicely, packed together around a nucleus, and it's just kind of, I don't know, very, can be boring looking, I guess. But the type of cells that we study in my lab are actually motor neurons, which are have a very extreme morphology. So in humans, they can actually grow up to several feet long. And the majority of the volume of these cells is not the cell body that's around and that contains the nucleus, but it's actually all these extensions that are axons and dendrites. And we're specifically interested in, so it has to regulate how genes are made from areas that are very far away from the nucleus. And we're specifically interested in genes that are made at the synapses that connect to muscles. So we have a technique to basically identify proteins that are being actively translated at synapses in these motor neurons. And this technique is called BONCAT, which stands for bioorthogonal non-canonical amino acid tagging. And basically it allows us to label and isolate newly synthesized proteins in vivo. So in living flies, we can do this. And the basic principle is that we introduce this mutation in our flies that allows for incorporation of this drug called ANL into growing peptide chains instead of methionine. So it lets us label newly made proteins specifically in motor neurons. And this is kind of a basic workflow of one of these BONCAT experiments where we can either isolate the cell bodies of motor neurons in the brain or the synapses at the neuromuscular junction. So in the top left, you have this good-looking fly larva that we feed this ANL drug that is a methionine analog. And then we dissect it. There's this cartoon of a dissection here where you have the brain up top, and then these lines extending down to the muscles are motor neurons, and they make synapses with the muscle. And that's specifically what we isolate, so we can get an idea of proteins made at the synapse. Some of the people in this Zoom call have helped me dissect these. And then we homogenize them. We do this click chemistry reaction where we can label these newly made proteins that contain ANL with biotin. So then we can purify these proteins and then send them to mass spec to identify them and quantify them. And here's some preliminary results from one of these BONCAT experiments. There's 3 different genotypes here, and we dissected 100 fly larvae for each of these genotypes. And this control regular genotype is just a normal fly that we fed normal food. So that signal is just background signal, basically. There shouldn't be any newly tagged proteins. And then these 2 genotypes are either just a normal healthy fly or this disease ALS model fly. So we study Lou Gehrig's disease or ALS because, for reasons that aren't totally understood, motor neurons start to die in Lou Gehrig's disease. And we see in our disease model fly that a couple genes associated with ALS, such as this HSC70-4 gene or phosphofructokinase, they're expressed differently at the synapse compared to a control fly. And what's exciting is that we've actually previously published papers specifically about these 2 gene targets. So we think this is going to be a very powerful way to identify genes of interest at the synapse in Lou Gehrig's disease, and also more broadly kind of show alterations in protein synthesis in our flies. And with that, here's my references and acknowledgements, and thank you.